As living things, cells have to grow and reproduce. That's what living things do. Let's take a closer look at the cell cycle, which is the process by which a cell is able to divide to make more cells. When a new cell is born, it starts in the G1 phase of the cell cycle. This is the initial growth phase. The new cell grows, it makes more cytoplasm, it makes more membrane, it makes more organelles, and generally just gets to be a bigger, more mature cell. When the cell is large enough, it goes into what's called the S phase. The S phase stands for synthesis, and this is when the DNA is synthesized. The DNA that's in the cell is copied so that now we have two identical copies of the DNA. This process, called DNA replication, only happens when a cell is going to divide. If a cell isn't going to divide, it isn't going to copy its DNA. Once we have two copies of the DNA in the cell, it's committed to finish the process of division. It goes into the G2 phase. This is another growth phase where the cell does all of the things it needs to do to get ready to actually divide into two different cells. Once everything is prepared for cell division, we enter mitosis. Mitosis is the actual part of the cell cycle during which the cell is going to divide into two different cells. Once that division is complete, we have two cells, and both of those are now in the G1 phase again. In this figure of the cell cycle, we can still see the G1 phase, followed by S, where the DNA is copied, and G2 and mitosis, all here in the inner circle. The outer circle shows us that every part of the cell cycle that isn't actively dividing, every part that isn't mitosis, is called interphase. So G1, S, and G2 are all lumped together into what's called interphase. And then we have the small amount of time during which division is actually happening that we call mitosis. Something else that's important to note is that while some cells do divide all the time, certain cells in your skin, for example, continually go through the process of mitosis to make new skin cells to replace the ones that are constantly being lost from the surface of your skin. Certain cells in the lining of your digestive tract are always making new cells to replace the ones that are damaged by the digestive enzymes. Cells in the hair follicles are constantly dividing to keep producing the hair. But other cells don't divide continually. Most of the cells in your body are not constantly dividing. They divide and then they're done dividing. The cells that are done dividing, we say, go into GO. At this point, they're not preparing to divide. They're not going to copy their DNA, and they're not going to go through mitosis. A good example of this would be neurons. Once a neuron is born, a mature neuron doesn't divide any longer. It goes into the GO phase. Cells spend the vast majority of their time in interphase, where they're not preparing to divide, but rather they're just existing as they are and carrying out whatever function they're supposed to be carrying out. But let's take a closer look at the process of cell division. Let's look at mitosis. A lot of work has to be done during interphase to prepare for mitosis. Remember that the growing, the copying of the DNA, and the preparation of all the various proteins and materials needed for division all occur during interphase, when we don't see that the cell is actively dividing. Once all of that is completed and the cell is ready to divide, we go into prophase. During prophase, the DNA condenses down into really compact chromosomes. Usually it's long thread-like chromatin, but that's really hard to work with when you're trying to separate the DNA into two different cells. So it contracts down and wraps up very tightly, coils into the chromosomes, the condensed chromosomes. And that's what we can see in prophase. When we stain cells so that we can see their DNA, we see these dark chromosomes all sort of clumped up in prophase. The condensed chromosomes have to be pushed together into a line in the center of the cell, and that brings us to metaphase. Special microtubules called spindle grow out and push the chromosomes together into a line in the middle of the cell. Remember that during the S phase, we made identical copies of all of the DNA. When these line up in the middle of the cell, one copy of each chromosome is going to be connected to one side, and the other copy, the identical copy of that chromosome, is connected to the other side of the spindle. Then we have a retraction that pulls these chromosomes apart, and that brings us to anaphase, where we can see the two sets of chromosomes separating towards opposite ends of the cell. This gives us an identical set of chromosomes at each end of the cell. We have a complete set of chromosomes toward one end and a complete set of chromosomes toward the other end. So when this now divides into two separate cells, each of the two cells that are formed has a complete set of the chromosomes and all the DNA that's needed to be a functioning cell. The last part of mitosis is telophase or telophase, and this is where we're actually going to separate into two cells. The process of separating into two cells is called cytokinesis. Actin microfilaments are important for carrying out cytokinesis. We form a band of these actin microfilaments under the membrane in the middle of the big cell, and then it begins to contract and get smaller and smaller and smaller. These actin microfilaments are almost like a belt that's being cinched tighter and tighter and tighter until it finally separates into two separate cells. It's nice for you to hear about the phases of mitosis, but I'm really only interested in you knowing the basics of the process. What happens in order for a cell to be able to divide is we have to copy the DNA, and then we separate the two identical copies of the DNA to opposite ends of the cell, and we divide into two separate cells. And that gives us two cells that each have all of the DNA they need to carry out whatever function it is they're supposed to do. It's very important that the cell cycle is properly regulated. We need cells to divide to make more cells when we need them, when we maybe need to repair a cut, or when we need to replace some damaged cells, or when we're growing to get larger. And all those times we need cells to divide. But we don't want cells to divide when they're not supposed to divide. Cells that are dividing out of control produce clusters of cells that grow and grow where they're not supposed to. And this is what we can refer to as a neoplasm, or, in more common term, a tumor. As you know, we usually consider tumors to be pretty bad news. Nobody likes to have a tumor. But there are lots of different types of tumors, and not all of them are bad. Sometimes we have some cells that are growing a little bit too much, and they do form a tumor, but that tumor is benign. Benign tumors are usually surrounded by some sort of a fibrous capsule. This keeps them separated from the surrounding cells so they're not interacting with them or spreading through them. And that's the other important characteristic of benign tumors. Benign tumors don't invade nearby parts of the body, and they don't spread to other parts of the body. When a tumor is able to grow into and spread or expand or invade into other tissues, or when it's able to spread to other areas in the body, that's what we refer to as metastasis. Metastatic tumors are more commonly known as cancer. They aren't contained within a capsule, and they do spread to other areas of the body where they can continue to further grow out of control and cause more neoplasms or more tumors.